Every plant, animal, and microorganism in our ecosystem functions as a physical archive of biological information. When we zoom past the cell membrane and into the nucleus, we reach the physical source of that information, the DNA double helix. Modern biotechnology tools translate these physical molecular structures into digital sequences, converting physical geometry into strings of readable code. This process generates 5 specific categories of data. We record sequence data from DNA and proteins, structure data mapping 3-dimensional shapes, and expression data showing which genes are active. We track interaction data between molecules and phenotype data documenting observable traits. This information originates from 3 primary sources. Experimental data comes from laboratory tests, public data is shared globally, and simulated data is generated through computational models. By capturing these measurements, a living organism becomes a repository of structured digital information, stored and analyzed far from the original biological sample. High-throughput sequencing technologies now allow us to read this biological code at speeds that were impossible a few decades ago. Every 24 hours, laboratories worldwide generate massive volumes of new digital records. A single experiment can replicate into millions of data points. Sequences, annotations, and measurements quickly expand, filling digital storage with unorganized streams of information. Researchers cannot find specific genes or compare protein structures if they are buried in millions of scattered, unindexed files. Without a method to categorize and search this output, the raw data becomes a digital landfill, too chaotic to support meaningful scientific research. The biological database was developed to solve this problem. It is an organized digital system designed for the efficient storage, search, and retrieval of biological records. These systems follow a 3-tiered structure. Primary databases archive original, experimentally generated data submitted directly by researchers. Secondary databases filter this raw data to identify patterns and classify protein families. Finally, composite databases integrate information from multiple sources, allowing searches across various databases simultaneously. This ecosystem acts as a filtration pipeline, organizing raw laboratory output into categorized, searchable knowledge. To see how a discovery enters this system, we can follow a single biological sample. After extraction and sequencing in the lab, it exists as a raw digital file. The researcher uses submission tools like BankIt or Sequin to upload the file. These tools ensure the data meets the technical requirements of public repositories. Once the database accepts the submission, the record is assigned a unique accession number. This tag makes the sequence a permanent, traceable part of the global record. Through this process, a sequence generated in one local lab becomes accessible to any scientist with an internet connection. If a researcher on the other side of the planet needs that specific sequence, they have to find it among hundreds of millions of other entries. They use retrieval systems like Entrez or SRS. These are sophisticated search engines that query interconnected databases to find exact biological matches. However, search results are only useful if the data is in a format that different computers and bioinformatics tools can read consistently. This is why we use standardized formats like FASTA. The first line is a simple identifier starting with a greater than symbol. The following lines contain the actual biological sequence data. These universal formats ensure that biological information can be exchanged and analyzed by different software tools across the global scientific community. Reversing the journey shows the complete pipeline, from the digital record back through the database to the DNA inside the cell. This workflow, from experimental storage to global search and retrieval, allows scientists to compare genes and proteins across different species and environments. A discovery made in one country can be analyzed and expanded upon by a researcher in another, creating a shared digital map of life. These databases serve as active engines of research, transforming raw, massive datasets into the organized knowledge required for scientific discovery.